lecture.
Tomorrow it is ready.
Actually, can you?
Moon?
Please check.
ঠিক আছে
It's okay. It's not on the screen.
9 to 11, then after that 15 minutes tea break.
Of course. Well, even every time, even 1 hour, it's too much for students. We'll take many breaks. Don't worry. But the screen isn't on. No, it's not on. My slide. Okay, now. Okay. All right. No, please, can you wait outside? You're late. So I'm keeping all the people who are late outside. You miss one lecture. Okay? Come on time tomorrow. Okay, so just wait outside.
Tea break is over because you not allow anyone to come back.
Please stay outside if you're late. Well, good morning. Okay, so all of you, so I've come a long way as professor here came from Japan, and you know, I was a student like you at IIT Bombay a long time ago, and so it was a, you know, the time was kind of different. We were students, we were taking classes, learning a lot of different things as engineering students do, but didn't have that much kind of opportunity to do research, learn, and widen the horizon that all of you have. Your curriculum is very different, and now, to the credit of Government of India, they are organizing these Gyan lectures. It's so amazing that they are spending money and time to bring people like me and others here, and giving you, as I heard, a scholarship to come, so you don't have to pay the registration fee to learn the opportunity, learn the topic. And I think it's an amazing opportunity that you should be very aware of. It's very uncommon. Most places in US, Europe, you spend a lot of money for such courses if they are offered, and they're very intense. Now, so here, my plan is that in about 1 week, I'm going to compress, please wait outside, 1 semester worth of material. That sounds too much, right? But you'll be surprised. We'll do it in a very nice way, and it'll be a lot of information, but through interactions and through quiz or questions, I hope you will learn the topic. Now, my plan, in fact, one of the students, some 2-3 students yesterday asked me, do I have to study tonight about brain because I don't have much background in this area? And I said, don't worry about it, although of course initially I joked. So based on that, I'm going to get started with some basic of neuroscience. Now, I understand the audience is very mixed. Some people have come from AIMS, they are physicians, and others are engineers. Some of you are young undergraduates, some of you are postgraduates. So we will create a very balanced way, and maybe a little bit boring for medical people if I go into basic, but then they can add some information, and if it's similarly technical, it gets too complicated, engineers can help the medical people, okay? And so we should make it very interactive. Someday you will all grow up, you're all grown up. It's impossible to give 20 hours of lectures. So I have to have a dialogue, I have to have an interaction. So I'm going to expect that, if not demand it, or if not incentivize it. Incentive is the best. Let everyone in. Please go ahead and bring everyone in. You know, the ones who are waiting, you can ask them to come in.
Okay.
I just wanted to convey a message.
Thank you.
so we are not starting off well because all of you have chosen to come late and be relaxed. So let's make a little global comment. If you go to Japan has what are known as bullet trains. They run precisely on time. I would expect that the classroom in Japan students will come precisely on time. I expect good students in US, since I'm giving a lecture at 9, will come here at 8 with a chance of interacting and having discussion between 8 and 9 to learn about something because you're curious, you want to ask questions. Many of you have chosen to come at 9:20. I think it's very disrespectful to the teachers. It's very disrespectful to the government, which is putting up and spending a lot of money to provide you this once in a lifetime kind of an education. And to each other, because we kept other people waiting, so we lost 20 minutes of time. So please be respectful of each other, respectful of your teacher, the incredible learning opportunity you have. So some of you have kept them out just to convey a message. So I really prefer that all of you are here at 9. I would welcome tomorrow, that how long will it take to walk from guest house to here?
So it will take 10 minutes.
Oh, that's too little. Anyway, I prefer that any of you like to meet at the guest house at 8 o'clock. We can walk over here. I don't want to be taken in a car every day and so on, so that we can have discussion. Okay, so let me just start again. So this Gyan lecture is an incredible effort by the government to, you guys missed the quiz. Sorry, we cannot let you come in anymore. Wait outside. No, it's okay. Come in.
But next time I'm not
Come in. Come in, please. See, look at it. So how disruptive this is. It's okay. Today it's okay. We'll convey the message and convey to each other. Some of the good students and the ones who came early should tell others. Okay. All right. We're going to keep this happening today. So tomorrow I will lock the door at 9.
Yeah, exactly.
Okay, so at end of story.
My appeal is basically the time is very important because the video streaming is going on, and then many people outside this room also attending, particularly the students outside this IIT Kharagpur also, and also it is streamed in YouTube and many others. So many people are attending these courses in fact offline. So may I request you to take your seat 5 minutes before the scheduled time. I have given the time, I have taken enough pain to prepare the schedule. Professor Thakor itself was very much serious about maintaining the schedule and others. So may I request you all so that you can take your seats at least 5 minutes before the time of the session starts. So today the first session, Neurophysiology, it will take around 2 hours or so, and then after that 15 minutes tea break. 15 minutes means is a 14 minutes 59 seconds. Okay, even not a 1 second will be extended. If you don't come within this time, then your room will be locked. Okay, please be serious about the timing and others. Thank you very much. Sir, please.
Okay, well, all right, so let's start now from the beginning, right? So good morning. Okay, if they're recording, then we can, everyone will be welcome, and we are here to learn, and we want to thank Government of India to make this kind of Gyan lectures possible, where people like me who come from overseas and alumni can give something back and in fact enjoy the interaction with some of you young students and creative people who have careers in mind, and vice versa. I hope you learn a lot. So in this almost, I think I have about 18-20 hours of lectures, so that means there's a lot, a lot of topics I'll cover, from, because many of you are engineers, maybe you want to know more about brain, and some of you are clinicians who need some engineering fundamentals. And all of you probably want to know what is the state of the art, what is new and exciting frontiers in this field. And so a little bit about myself. So I was an undergraduate student of electrical engineering at IIT Bombay once upon a time. And then I find it very disturbing. It's again, some more people are coming. So from there, my undergraduate BTech project, which is what made change my life, was to develop a electronic circuit and other devices for monitoring heart rhythm. And I continued that to a PhD in arrhythmia, abnormal heart rhythm monitoring at University of Wisconsin with Professor John Webster, whose book is very standard and popular around the world. And from there at Northwestern University, I became a professor of electrical engineering to develop implantable electronics. And then for 35 years, I was at Johns Hopkins and still am affiliated there, where I've done research on heart, brain, prosthetics, and many other topics. And then last 5-6 years, I direct a brain institute in Singapore, which has a wide range of neurotechnologies being developed in research. So this course is kind of based on the material I teach there, as well as research that we do, and more general topics. So in one way, I was a young student like you once upon a time. I haven't forgotten. My children are probably around your age, so I haven't forgotten that. On the other end, I'm a teacher, and I want to give you good knowledge, but also good discipline, good principles. I want to give you a lot of information, but I want to also make you curious and excited about these things. So to do that, let's make it a partnership, which means that I will present, you can ask questions, make it interactive. I cannot do 20 hours of monologue. If you don't do it, I will ask questions. And can I get that bag?
Yeah, sure.
Just give me nearby. So today many of you came late, so that was not very pleasant for me because it disturbed the flow, and it's not respectful of the people. So we have decided that you will absolutely precisely come on time tomorrow, and everyone should be settled 5-10 minutes before 9. And anyone late, anyone late tomorrow will not be allowed in the class. So from this tomorrow on, let's have in fact, subsequently, we should have that discipline even during that break so that we can cover material, plus look at this disturbance doesn't happen. Now, incentive is better than anything else. So I brought in incentive. So this is a bag of chocolates. So whoever gives good answers get a chocolate. Can you please help open it? So there will be that. It's a small incentive. We could try money if you like. So let's start with the first chocolate. How many brain cells are there in human brain? What is this? He wants a chocolate right away. But let's try a student. If the students fail, then you can.
2 billion approximately.
Yeah, you raised hand, so please speak up.
It's billion or million, sir. It's uncountable. I mean, 12.
Some 3 trillion.
So the 100 billions of nerve cells are there.
Good. Anyone want to do a little better than that?
In adults it is 100 billion, and in at birth it is 1 trillion.
I don't know. So let's just talk adult brain. A little better than that. Okay. So round number. Oh, sorry. About.
Souvenirs.
Okay, fair enough. Souvenir for you. Oh, sorry. About 80 billion, but round number to 100 billion. Does having more neurons make you smarter? Okay. Well, there was an interesting comment made that at birth you have more neurons and they get pruned to a smaller number. And then, and that is why use it or lose it. If you don't activate and engage your mind and brain, you lose neurons and synapses. Okay, so we're off to a start, all right? So, anyone has seen a neuron? So this is it. So this is a growing neuron, of course, in a micro dish, a culture dish. So now why did that happen? I'm a neuron too. Okay. So this round thing is the cell body, soma, and those little long thing that's going on can be called neurite because it's artificial, but it's really otherwise made up of axons, which is like a long cable that takes a message to the body, and dendrites, which are these small tentacles that go out. And in fact, these very small tentacles are what are called growth cones. They are trying to find other neurons to connect to. A neuron connected to another neuron forms a synapse. So this is the essential building block. Kind of for engineers here, this is your transistor. And with its own wiring, and when the transistor connects to another transistor, in this case, a neuron to neuron is a synapse. Okay? There are more complicated structures, part of neurons, we'll learn about it. And in fact, what we want to learn is from this neuron, how can we make even an actually artificial brain? And we can get to that some point. So this is another picture. This is how neurons, if the video works. Yeah, so these are, you see in a microchannel devices, these neurons are here on the far left side, and these are long wiring. These are the axons growing. And you can see these little growth cones, these little tentacles, they are out trying to find other neurons, and that's the entire neural network that how forms in the brain. So think of this as the developmental part of neuron. So when brain is developing, cells will grow and find the connection network. Adult brain, the wiring is fixed. So then a lot of changes occur at synapse, which is neuron to neuron connection or a neuron to axon connection. And this wiring is two parts. So one is the cell. That is what is in the brain. We call it gray matter. And those long little wires are axons, which is the white matter. And that's what our brain is made up of. So we are beginning with the fundamentals, alphabet of brain or neuroscience. Okay. So today I want to do a quick recap of the nervous system. All right. So that everyone can be become familiar with the vocabulary, and some of you may know more. I strongly encourage to participate, maybe share some information if you like, so that it will be more interesting. So let's now go into the basics of a neuron, right? So this is, as I said, this is the soma, cell body. Always cell body has a nucleus. And then it sends out these little dendrites. These are little wires, tentacles go all over. And then there is this long tail-like axon, which is a big messenger that takes the message too far away in the body. For example, an axon can, from the neuron in the spinal cord, has an axon that's going to go all the way to your muscle and then make the muscle twitch. So message is coming from neuron, move, and to that message is relayed via axon to the muscle fiber. So this is the way that neurons work. There are these axonal terminals, or for the matter dendrites. You can form synapses, which are connection to each other. The axon terminals can connect to the muscle cells and form neuromuscular junction. Question. What is the longest axon? Yes? What is the longest axon? So just pause and answer. The longest, or where is the longer axon? So spinal cord itself is where the cell body is. So the cell body is in either in your brain or it's in your spinal cord. So where from the cell body, we have some more people coming in. We are not going to admit any latecomers from this lecture onwards. It's extremely disruptive to all of us. So it's quite unconscionable actually when you think about it, that you coming in at 9:30 on a first day of a class. I'm really shocked. Okay, so what is the, that's a very common sense, very simple answer. Exactly. So longest could be from spinal cord to your leg. So you can say maybe 2 meters, 1 meter, 1.5 meter, right? So axons can be quite long. And dendrites are small tentacles going all over. So incredible massive network is present. All right, so quiz question, just for the sake of changing. How many neurons are in the brain? Okay, latecomers, blah, blah, blah. There were trillion, quintillion, big, small, forget it, about 80 to 100 billion. You know, it's the same way the human genome program was going on. People say, how many genes in the in cells? I mean, in our biology, how many genes?
21,000.
About 28,000. Yeah, so good. So it's not a large number, right? Small, right? So similarly, when you think about it, 80 billion is not a lot, right? I mean, it's amazing considering that if you think of today's computers, that we do so much with it. Just for fun of it, can anyone name a Nobel Prize for brain research?
Mayors couple for 2014 or 13.
Right, so we'll come back to that, yeah. Hodgkin and Huxley, wonderful. We're getting started, I'm glad. Sorry, I can't give out chocolates too quickly, but I'm very tempted. Okay, so if you never get a chocolate, come back to me, because I have a limited supply.
Not exactly a Nobel Prize for brain research, but the person concerned is presenting the brain research, Professor Tomegawa.
No, he did get a Nobel Prize, so it was not exactly, you're right, that Tonegawa's work was not directly into brain research, but it has implication at a molecular level.
Yes, but he's presently into neuroscience.
Yes, indeed. Okay, very good. So let's have this kind of slide to manage time, side discussion afterwards, but wonderful that you have such extra knowledge. So you know, in this class, we'll go from very basic to Nobel Prize. Why not cover that? And indeed, Hodgkin-Huxley name will come up, and some other things about memory and learning will come up as well. Okay, so let's do a quick recap of the nervous system. So I already showed you the neuron. So that was like synthetic, artificial in the sense that it was in culture on a micro channels, microfluidic system that is, so it's very, you know, it takes a little bit artificial from. If you actually really go into the body in 3D, I mean, you can see that it looks fantastic. I mean, it's a full 3-dimensional architecture. Actually, one on the right, kind of representative of axonal fibers and dendrites and so on. And so the one other very important number that you're getting, neurons connect to other neurons, and that connectivity is incredible. Every neuron might make about 1,000 to 10,000 synapses. Therein the difference between computers and the brain. Because, yes, we have transistor, but one transistor connects to 2 neighbors, 3 neighbors, maybe if it's really cutting at 3D even then. Here, 10,000 fold. So now you can imagine the number of synapses are almost like 10 to the power 14 or 15, and they are plastic, they are adaptive, they can change, and our transistors don't change. So therein lies a number of uniqueness of the brain and nervous system, which is why brain is so amazing. Then if we go into a little detail, wow, I mean, can you say wow? Show expression, guys, you are dead boring people. I mean, this is the brain. Can you see that? It is mind-boggling. So on the left side, by the way, the color is artificial in the sense there are certain dyes used to make it synthetically colorful. So they are called fluorescence dyes to give a color. So if you excite with certain light, you get this color. So this is what is called green fluorescent protein and makes it green. So you see the cell body, and you see these white tentacles going up. Those are the dendrites that are spreading, and they will go up. So this is the cortex. This system is the cortex, and this is one layer of the cortex where all the cell bodies are. They are sending out all these axons. Dendrites that are going up all the way, and then they are spreading all over and going back and making connection. Of course, they make a lot of connection here as well, and this is can be an entire course in neuroscience, but you can see the complexity in packing. And by the way, not every single cell is eliminated by these techniques. They are selective. In fact, one of the earliest person to develop a stain, a guy called Golgi, got a Nobel Prize. And for the matter, Cajal, who did this kind of mapping, who is the giant of the neuroscience, he also got a Nobel Prize. To develop these stains so that you can actually identify these neurons in cell body. And this is the three-dimensional architecture at the current cutting edge people are trying to unravel, from micro to even nanoscale using different new technologies. And the right side starts to give you a really very good three-dimensional architecture. Of course, I wish if we could show this in stereo, and it's a technique, it's called rainbow. It's a way of colorizing different types of neurons differently, so it gives you the diversity. And then, you know, like we like to make our computer in 2D, 3D with geometric structure. This is very complex 3D, although there is such structure like this to some extent, which is helpful, but there is this kind of mess of complexity probably in this area. By the way, you see a little thing. So again, see the dendrite, you see really tiny dots, really little, tiny little things on it, and those are where the synapses are forming, the synaptic butons or that. So every one of those little tiny dot that you see on each of those dendrite is probably another dendrite that's touching and making a connection. And of course, because it's selective staining, if you did everything, you wouldn't see anything. It will be incredibly complex, although you can look up some electron micrographs and it can show more detail. So I hope you have some deep appreciation of incredible beauty and complexity of the brain, right? This is just the beginning. And I hope you have some vocabulary now of neurons, soma or cell body, dendrite, axons, synapses, and so on. There's a lot more to it. There are many different types of cells. Yeah.
What is the cortex?
So we'll come to that. It's the brain. The cortex is the brain, usually associated with the outer layer. The bigger part of the brain is what we'll call cortex. So now, to answer your question in some way, we moved from neurons all the way to, let's say, nervous system. So entire nervous system is, of course, a incredibly complex maze of development. To begin with, it's the brain. So we think of the brain, which is what it's the large brain, and then there's a small brain, or what is called cerebellum in the back, and then this is the spinal cord, and then these are the nerve fibers. They go throughout the body. So now we have to think of the entire brain with its very complex heterogeneous structure, but systematically now we have found different areas of the brain to a great extent specializing in different things. For example, there is an area of brain called hippocampus, and generally being known to associate itself, you know, with memory. So there was a remarkable research about maybe 50 years ago, where there was a patient who used to get a lot of epileptic seizures, and so they cannot function or something. So surgeons had this idea that the seizures arose from this part of the brain. So they just removed that part of the brain called hippocampus. So when they removed that part of the brain, that person could form no new memory. So everything before that surgery he knew completely. Anything after that he would not form the memory. So now you can tell him that neuron is made up of soma, axon, and dendrites, and okay, and the brain is this. Oh, what is neuron made up of? He doesn't know. But in childhood he lived there and went there and ate this food on that day on his birthday, he will remember. So this person, HM, his initial, became very famous because it gave us an idea that brain has certain organization and that certain specialization, and maybe something that fascinates all of us. What is memory, or where is memory? It's one answer. It's an answer to memory is very long. When are we going to discuss all these deep questions about learning and memory? Anyone has answer to that? So I said when. No. When? What time are we going to discuss all this? I gave an answer a little bit earlier. At 8 o'clock, as we walk from guest house to here, we can talk about these things, because lecture room is a boring place to give a lot of facts. But we can talk about, you have curiosity about where is memory. So is there hippocampus, but what about this? But I heard that memory is, you know, Eric Kandel got a prize, a Nobel Prize for memory, doing this. So we can have those kind of dialogue, or perhaps even in the evening, if they don't take me away for some meetings, then we can have this kind of discussion, or over coffee break, right? Although these are short, so we will do that kind of discussion, because there's so much topics you may. So I want to make sure that we can have these questions and discussion of things that interest you. I won't be able to answer everything. Then we'll go and do some research and find out. Okay. So let's go back here. So the brain is made up of this, or nervous system. Let's call it that way to be accurate. A nervous system is both all cortex, the brain that you see, the spinal cord, and the nerve fiber. By the way, there are two types of nerve fibers. So they are on the right side. If you look at the picture, they are parasympathetic and sympathetic. And these parasympathetic and sympathetic are nerves that control. So first of all, they are called autonomic nervous system. So let me back up a little bit. The nervous system is both systemic, something that you think and they can relay the message and makes things happen, like moving my arm. It's very voluntary. And therefore, these systemic nerves are voluntary control of sensation, motion, etc. My vision, my hearing, this is all relayed by these nerves. On the other hand, these autonomic nervous system is not something you can control. So these are like your heart rate, your blood pressure, your control of kidney and liver, you can't control. Something like bladder you can learn to control, children cannot, little infants. So there's an incredible architecture of nerves throughout the body, and they are connecting and making muscles move. They're providing sensations to our eyes and ears, and these nerves are also controlling our life, our living. Sympathetic nerves excite, so to say, so my heart rate goes up. I want to start running, I get a sympathetic tone, heart rate went up. I ask a very stressful question, how many neurons in the brain, or exam question, the sympathetic excitement goes up, certain chemicals are released because it creates a stress response. And then there are relaxation. Yesterday we talked about meditation. So in meditation, your parasympathetic nerves are relaying so that you can relax, and muscle, nerve tone, the heart and rhythm reduces. So there is a wonderful balance of sympathetic and parasympathetic in the nervous system. You can see again how beautiful the whole nervous system is, how amazing the control architecture is. Obviously, I have so little time to cover each one of those topics. But you can see that it shows that there are so many different organs are affected by these nerves, as very obvious that our arms and legs are affected. Okay. So what is the biggest part of the brain responsible for intelligence and cognition and things like that? So let's try this. I'm glad that you're all participating. It would be kind of a little bit logical for me to go to one person who raises a hand and so on. So you said cerebrum, and so it's good. So that way, what happens is that the cerebrum, and particularly our frontal lobe of the brain, in that picture that you saw earlier, this is where we more associated human-like cognition, intelligence, and so on. It's the so-called prefrontal lobe, and that's where a lot of the higher brain functions reside. Of course, the brain functions are distributed throughout the brain, and there's a lot of distribution and overlap, but certain specialization occurs. So for example, my back part of the cortex is more affiliated with vision, and temporal side of it is more affiliated with speech. How do I know that more likely the left and right is for speech? How did they find out?
By implanting electrodes and seeing the activity of the various parts of the brain.
Yes, so that's a interesting answer. I'll repeat if this is being relayed or recorded, that yeah, you can implant electrodes and stimulate the temporal lobe, and if something happens to your speech, either it interrupts it or changes it or something, hey, it's something to do with speech. But if you implant in the visual cortex, that will not happen. So you know that there's some specialization. Somebody?
As the stimulation
Yeah, but actually that came later. How did they find out?
By removal of the
By removal. So somebody has a brain tumor, that part of the brain is removed, suddenly loss of speech. My mother-in-law had stroke, which I will talk later, and then afterwards a little recovery, etc., and at some point she fell and hit her head on the bed, and there was a real trauma. I believe it was on the left side, but a real trauma to the head, and she's aphasic. She cannot speak. She can understand, she language, she types, she does everything else, but cannot speak. It's called aphasia. So brain has such specialization. At the same time, brain has plasticity to overcome such problems. So there is a high level of organization in the brain. And so we know about what part of the brain is responsible for memory. Quick! Okay, sounds good. Huh? Okay, so that's where it's a reasonably good point, right? That it's really important to understand that something like memory is multiform, multifaceted. There's semantic memory, there is spatial memory, there is, all kind of memories, right? So the hippocampus is where the spatial memory, the physical geographic localization. When hippocampus removed, how did they find out? Well, this is the lecture. Can I say someday you can say I heard about more Nobel Prizes in this one lecture than in my entire life, because that's where somebody recently got the Nobel Prize. I think one of you said 3-4 years ago, because they studied this hippocampus, putting microelectrodes in that, and rats that are running around a room, they find that certain neurons are active when the rat is in this corner, and some neurons are active when it's in this corner, and some neurons are active when the rat is in this corner, because the rat goes around trying to find food right, and makes a map of the place, kind of like GPS. So when the Nobel Prize came, they advertised it as GPS of the brain. So it's done long time ago by people in one scientist, O'Keefe in UK, and then a husband and wife team in Norway, Mosers. So they studied. They put microelectrodes and say, let run around, and they found that only certain areas brain cells were active, which means that neuron was remembering, encoding location. See, we all have GPS, right? We know on the campus, if you go here, then that's a computer science building, and now I know the direction to go there, and we have created a map. Well, rats are creating the map, and the hippocampus plays a very active role, but it's just that one kind of memory. There are a lot of other memory. They're very distributed. There's a kind of three-dimensional spatial part of memory, but there's memory also at synapse level, and then even at molecular level inside the neuron. But for now, we can give at least a simple answer that hippocampus is at least some type of memory, like I said, spatial memory. All right, what happens if spinal cord damaged? What happens? So start with something, keep it simple, stupid. Everybody knows what a kiss is? Keep it simple, stupid. Okay, so give the simplest answer first, then we can get more complicated. So paralysis, now you added something? So reflexes, fair enough. I mean, we touch something hot, that oops, the spinal cord plays a very intelligent, active role. Message doesn't even go to the brain. The spinal cord is the reaction. Okay, so what happens when the spinal cord gets damaged? Well, let's ask the other question. Can it be repaired? No. Rewired?
It depends upon the damage, how much it is being damaged. It can be termed in terms of classification as partial, complete spinal cord injury.
Okay.
it can be.
All right. So you guys are the medical type, but I doubt it. I question it. So spinal cord is part of the central nervous system. The peripheral nervous system, the nerves, do regenerate. So regeneration means that it's damaged and regenerated, kind of like lizards regenerate their tail or something, but in this case nerves have a natural tendency to regenerate. But central nervous system, whether it's spinal cord or cortex, which is part of it, doesn't regenerate. So it cannot produce new neurons. Repair, plausibly, right? I mean, a repair means many different things, biochemically, molecularly, and so body itself probably does a repair by reassigning the functions. And it's a more complex part of biology, but I wanted to convey more of a message that central nervous system doesn't regenerate and so on, but it can learn to. So people who get stroke, for example, where some part of the brain gets damaged, other part of the brain will take over. However, and same thing in spinal cord, if there is a partial limited damage, you can eventually learn to walk because there is a plasticity and it can, but it doesn't rewire per se in the sense that somehow wonderful new wiring takes place. But as those fibers regenerate, they try rather unsuccessfully. So when these spinal cord fibers are trying to regenerate, repair, they don't find the target. They don't repair very easily. It's one of those really hard problems. That's why people with paralysis, they can improve their some level, but if the complete spinal cord transection cutting, you cannot come back, right? So Christopher Reeves, anyone remember who was that? Superman, Christopher Reeves, and you cannot walk. And so this interesting area of research related to repair, rewiring, regeneration. As engineers, what we can do is to stimulate the spinal cord as a pseudo artificial way of restoring the function. But if a nerve gets damaged, yes, the nerves have a natural tendency to repair, regenerate, and therefore that is realistic. Although rewiring and all of that to get it precisely targeted, very difficult. It's a very complex area of research in biology to teach nerves how to find their goal and targets. Okay. All right. So let's now switch gears to diseases of the nervous system. This is like our doctors here, entire course in neurology in 10 minutes. So a little bit challenging, but we can at least do the vocabulary. So everybody knows about Alzheimer's, right? So what causes Alzheimer's? Age doesn't cause it, but yes, people who are older. Sorry, you have to either raise a hand, speak louder.
Degeneration of the structures of the brain, cortex.
Sort of.
Due to which that shrunken brain appears.
That's the outcome. So the deep late stage Alzheimer's, the brain may seem shrunken. Somebody else? Yes?
The connections get, the synapse, those connections get broken. So
Not necessarily in Alzheimer's, yeah.
Hyperphosphorylation of tau.
All right. So first of all, I asked what causes and nobody knows. But Alzheimer's is associated with something, so it's a coincidence that anyone who has had Alzheimer's seem to show something, and that is this tangling. This word tangles, dying neuron with tangles. So there are these protein fibers and so on. So you mentioned the word tau is a kind of a protein. So these neurons, and they start forming tangles. These undesirable protein or plaques start building up. So Alzheimer is associated with plaques. Alzheimer is associated with these tangles like tau protein and so on. It's not, we don't know that it's a cause, it's more likely an effect. We don't know what the cause is. Can you diagnose Alzheimer's? Or if yes, then how? If no, why not? Chocolate. If you're guessing, then no chocolate. Because Alzheimer gets thrown around a lot. Everybody talks Alzheimer's. My grandmother had Alzheimer's or something. You're more likely than not. Oh, sorry. Go ahead. Go ahead. Yeah, always.
Alzheimer's can be diagnosed on autopsy.
So Alzheimer. Here's a. That's not fair. Okay, I need an engineer to answer. So next time when you answer a good engineering question, that time you get this. I'm going to run out. You guys are rather smart. I'm going to run out of chocolates. I don't have that many. I mean, one per student. So it's true. So in a sense, the answer is that everybody kind of talks about my grandmother, somebody else, Alzheimer's, and so on, and that's not true. In the sense, more likely than not, it's what's called dementia, just age-associated mental decline, maybe because of loss of brain cells, blood vessels in the brain become clotted, various other things happen. So it's a degradation with age. However, Alzheimer is a unique thing. It is these plaques and tangles and so on, and they can only be confirmed on autopsy when the brain person recover, is dead, you do a post-mortem and you say, whoa, that person had Alzheimer's. So, that is the confirmation. However, you get a lot of pseudo research, in a sense affirmation predictive, that brain images look different. People try to look at EEG and say I can predict Alzheimer's, and these are in small samples. People try to make these predictions because it is not possible to go into the brain and do the imaging because of the skull. So, it is not easily verifiable. Okay, but you can see the difference between an shrunken brain, tangles, various changes. So, Alzheimer's is one of those grand challenges. It is affiliated with aging, and that is why these diseases are coming up, because people are living a bit longer. But it does raise a lot of questions or research on how to detect Alzheimer's, how to confirm it, which is by far the most important question right now, because we can hardly treat it if we can't even detect it. And if we can do some early detection, it will be very helpful, because maybe we can develop some way to cope with it. Maybe some memory exercises and certain preventive behaviors and so on, so prolong a good active lifestyle. In developed society, economically prosperous, where people are living, in a country like Japan, I mean average life expectancy is now in mid-80s or even 90. It is going to be a profound, serious problem, a tremendous burden on the society and cost. So, this is one of those grand challenges.
So
yes?
some researchers have developed a sniff test, what they say.
Yeah. So, fair enough. So, what happens is that he said that people have developed a sniff test, and I think what you will find by and large is that there are a lot of these kind of reports that, because people do want to detect things, and when I say that these are pseudo or emerging, meaning they are not, for you physician, equivalent of saying that by studying 50,000 people in 50 countries, getting absolute affirmative data and publish in New England Journal of Medicine, then which is the top clinical journal, then I guess that is confirmed. I mean, as confirmed as it can be. Otherwise, yes, sniff test, behavioral test, this scan, this image, change in EEG, there are these low level studies where everybody wants to detect Alzheimer's and this and that, and these are not very rigorous and robust, and you can't say, oh, grandpa didn't sniff, oh, he's got Alzheimer's, okay, go away, you can't do that, right. So, I think the point is that there is a tendency to jump to grand conclusion, anything about brain diseases, and good science, good engineering needs to be rigorous, robust, and Alzheimer is one of the most enigmatic ones. Parkinson's disorders. So, anyone know what is Parkinson's? What happens to patients who have Parkinson's? Somebody else? So, yeah, so tremor or movement, tremor is the most obvious symptom. What is the other symptom? It's written there.
Bending of back.
Well, yeah, and more likely the one on the lower side, the rigidity and shuffling. So, the Parkinson's, so most prevalent way of looking and thinking of Parkinson is the tremor, the arm. If you try to reach, your arm will show oscillations or tremor. But Parkinson's patients also show a degree of rigidity and shuffling gait, or sometimes freezing, like they're completely cognitively intact. They were walking and they just freeze. They won't move. Remarkable. Think about how a brain would work like this, where you just can't move, and if you give a little nudge, the person will start moving. So, there are these lot of behaviors like this, but it all arises because there is a loss of certain brain chemicals. So far we talked about neurons and all of that, and then neurons have electrical activity and things that we will study, but there is also chemical activity of neurons, and there is a chemical called dopamine. And when it gets lost to a very large proportion, there is a certain very special area of the brain where that motor movement control is. There are area called basal ganglia, and there are certain regions where there is a neural circuitry for controlling movement. And when that movement, when neurotransmitter level goes down quite a bit, like 60-70%, at that point these come up. So, Parkinson also unfortunately is detected rather late, by the time there is a lot of loss of neurotransmitters. Fortunately and incredibly remarkably, there is a treatment, not a cure, but a treatment, that stimulation of that area of the brain at certain frequencies, the tremor go away. So that like it's remarkable, and so that is there is now a kind of like a therapy or treatment for people who are Parkinson's. There are many diseases of the nervous system. This is one, I would say, it's called multiple sclerosis. So MS also is a disease of an aging brain, more likely, but again it's more vivid later in life, and it has to do with myelination. So what is a myelin? Myelin is a protein. You remember I told you about what a brain cell is made up of: soma, then axon, and myelin is the insulation around the axon. So therefore, then myelination, when it goes away, so that insulation makes the signal on the axon go fast, and so many axons are myelinated, and therefore they rapidly transmit the message. MS is a disease of that demyelination, that insulation goes away. Think of electrical wiring, right? If no insulation, you get short circuit, but something like that. So there is a, and therefore there is a progressive decline of the movement and even cognitive functions. No cure. You know, these are tremendous emerging health problems. So if somebody gets MS, it's a progressive degradation of their nervous system. What is this? Who is this? So he is symbolic of this problem, and that is Stephen Hawking is potentially Nobel caliber physicist who has discoveries in black hole and so on. Incredible, right? Incredibly is kind of like as synonymous to genius as you can be. So the brain, unlike the MS and other things or Alzheimer's, where brain, in Alzheimer's the memory is lost, MS there are cognitive deficits develop, Parkinson's there's a motor and tremor function change. In ALS, amyotrophic lateral sclerosis, or it's also in US called Lou Gehrig's disease, there was a famous athlete who was amazing, and then he started to lose the function and said, wow, how can such an incredible athlete lose the function? So in his case, you can see that he doesn't have the motor control. His brain is completely intact, but it doesn't have motor control. And so ALS is also progressive, and in some situation people can get into what is called locked in syndrome, where the brain is completely intact but no message is going to the rest of the body. And that is basically was the trigger, catalysis for kind of things we want to do here in this class, which is things like brain machine interface, because a person who cannot, so I'm going to have my laptop shut down. Okay. So, you know, you can see that all kind of brain disorders have started to, you know, come up when as people have aged in an aging society. What happened to my cable? Oh yeah, can we, you know, I don't know if it has got discharged completely. That is really strange. Well, hey, you know, this is a good time to take a break, 5 minutes, right? So in whether matter of brain or anything else, and yesterday I met up with the director of IIT Kharagpur, and we were talking about learning. And one of the biggest fallacies of teaching and learning is how much attention we can provide. All of you, so most of you are students. How long can you be attentive? 40 minutes? Are you serious? What's the correct answer? 20. About 20 minutes, yeah. So I went well over that time, but I managed to keep things going. Was, I guess, less boring, so maybe a little bit longer. So this is a good time to talk to your neighbor and exchange, say hello to each other, introduce yourself, because you could become friends, collaborators. Hodgkin and Huxley, you know, Hodgkin, Huxley, they collaborated, got a Nobel Prize. Sackman and Nair, a physicist and a physiologist. Sackman was a physiologist, and they got Nobel Prize. So talk to your neighbor for a few minutes. Please talk to your neighbors. You know, they are people, nice people. Say hello to each other. Say hello. Okay, I don't know what happened. Suddenly it's shut down. Ah, no, it's okay. But something happened. This is really bad. Okay, good. So everybody is awake, alert. Nobody has Alzheimer's. It's all good, you know. Otherwise, suddenly, you know, and obviously I could see some of you a little bit drowsy, so I can tell all that things. Okay, so good. So ALS is one of those disorders. Epilepsy is one of the other ones. And epilepsy, on the right side, later on we'll see different types of brain rhythms. Some of you are working on EEG, but you can see that epilepsy is a brain rhythm where large level of oscillations are seen. Think about our brain. Are 80 billion neurons doing anything collectively together? No chance, right? Right now they're all doing, I'm moving my arm, I'm speaking, I'm interacting, and so there's a lot of seemingly chaotic activity. There's, of course, there's coordination, but by the time you include, it's like when you guys were, remember, all of you were talking, it sounded like noise, right? From far away, because while you were talking one to one, from far away, when all the voices got merged, it became noise. So the brain is the same way. When millions and billions of neurons are working together, when you record brain rhythm, it looks like noise. But in epilepsy, it gets into these oscillations, and therefore these oscillatory rhythms affect a person in many various ways. Certain types of epilepsy, which is what people generally think about like movies and melodramatic, is called grand mal epilepsy, in which case you get a complete, you know, convulsions. A person can collapse and all of that. That's an extreme version, but there are many other epilepsies where you just get some small seizure, so something called absence seizure, where you just don't even move, just freeze. And it's just a basically disorder of a brain rhythm. A very interesting area for engineers in neurotechnology to how to detect epilepsy. Lot of research papers, lot of PhD theses on how to detect epilepsy, and a little less so, but emerging, how to treat epilepsy. Of course, you can treat by drugs, but nowadays with certain devices to again stimulate the brain or nerves. So Alzheimer's I talked about, how did that come later, epilepsy, brain tumors, right? Cancer occurs, and tumor of the brain, one tumor called glioblastoma is extremely lethal. Within 6 months a person could be dead. And so brain tumor, how do you detect brain tumor, how do you image it, remove it, you know, can be a profoundly important in technologies type as well as research. Sorry, I didn't have a slide on stroke, but it's, I mentioned earlier, right? Stroke occurs, why is there a stroke? What causes stroke? You answer too many questions, I'm not going to bother with you. Somebody else, somebody who has never said anything. Chocolate, come on, incentivize. What causes stroke? But if you give a complete and correct answer, I'll come back to you. Come on, I mean, this is the best chocolate in the world, some billion chocolate. You can't, money? I don't know. 10 rupees? It meant something in my days. Today it's an insult. Okay. All right. What causes stroke? Doctors can speak up. What causes stroke? But I want complete answer.
Occlusion of the blood vessel by a thromboembolic, or
You know, you guys engineers, next time you speak in a complicated way. You know, the quantum electromagnetic Maxwell theory of that. You know, so that way he shuts up. Okay, sorry. Repeat so I can explain.
the blood vessel lumen gets occluded by
Here again he used the word lumen. I mean, why doesn't it say, keep it simple? Yes, blood vessel lumen, right, he's accurate. Go ahead.
occluded by a clot which could arise there itself. or it could be originating from elsewhere in the body.
Yeah. So, you're not still going to get chocolate. So, blood vessel, and lumen is the inside part of the blood vessel, which is usually nice and smooth, but eating not too many chocolates, but eating all the Indian gulfi, ice, whatever it is, sweets, jalebi, then too much ghee and all that, and then it gets clotted up, or the clot can go somewhere else, from somewhere else, and go up to some part of the brain and blocks the blood vessel, and suddenly no blood supply. The brain cells die. How many minutes and they are dead?
1 minute, 2 minutes, 5 minutes.
About 4 or 5 minutes, yeah. So, but it's a progressive, because no energy, no oxygen, no glucose, etc. But why am I not giving you chocolate? Because you only gave half the answer.
It can also be bleeding event, hemorrhagic stroke.
So, that is you know, just so that you educate as an educator, not so much because he has studied all that. So, I don't think it's for smarts and intelligence, but knowledge, and he educated, right. So, other one is hemorrhagic stroke. It can occur because of bleeding. The blood vessel pops because there are certain times malformation occur. And what happens in stroke? What are the symptoms?
Commonly, it is one-sided weakness,
Mm-hmm.
inability to speak,
Mm-hmm.
or a loss of sensation in one half of the body,
Mm-hmm.
as well as other forms of strokes can also be present. Patient cannot recognize objects, cannot write, cannot read.
Can be amazing. There can be brainstem stroke, and you can drive. There's different part of the brain that's important for breathing and all of that. But most likely, it's visible in a motor sense. You get one side paralysis, movement. I think there's a 4-letter jargon word. You should be researching, telling me so that quick, fast, exactly. Very good. Thank you. You guys can split that chocolate because I don't know how, you know. Because I owe you one, I'm going to remember that. But fast is so
Fast
face.
means the F is the face, that face on the opposite side of the stroke gets paralyzed. Then is the arm. So the arm on the affected side doesn't get up. And then is the speech. S is the speech, and T is the time that you need to be taking the patient within 30 to 60 minutes to the hospital.
A little bit more. I'm going to take a piece of chocolate. 30 to 60 minutes is too much. So, you got about 3-hour window. So, what happens is that fast is a way, guys, all of these, something someday will happen to some older senior family member. So, you should know that facial droop, slurring of speech, movement, language, all of that will affect that, and these are symptomatic. The time is limited window. The reason is that these kind of occlusive stroke in particular, hemorrhagic is a serious problem, that it will just bleeding and there is much damage. But occlusive stroke, there are now clot dissolvers. They can dissolve the clot if you get it to the person within about 3-hour window. So, it's almost impossible to get within an hour, because by the time you detect, go to the hospital, then they have to do a CT scan, X-ray, brain image to make sure it's not bleeding. And after all that is verified, they give this drug called TPA. Then you could dissolve the clot and a person could just be fine. But if you don't, then you got permanent brain damage. Okay? So, stroke, there are many such problems. Back pain, inflammation, headache, migraine, these are all, you know, these are all part of the nervous system. So, you see there is a lot goes on in nervous system, that is great, works well, and when it doesn't, you got these disorders and disease. So, how would you treat Parkinson's disorder? By stimulation of basal ganglia and or some substantia nigra. There are certain regions of the brain by applying some high frequency electrical stimulation. Yes.
So could you have the neurotransmitters present in the synaptic cleft for a longer period of time?
Yeah, well, so as you heard that Parkinson is produced by loss of dopamine. So, there is a drug called L-Dopa. There is a drug that's given as a dopamine precursor, which kind of replenishes the drug, the dopamine level, and that works up to a period of time until the brain or the body gets habituated, then stops working. So, in fact, most common standard of care is a drug to replenish. Now, we can't go down to every neuron and synapse, and there are 10 to the power 15 of them. So, we can't go to those. So, basically it's the drug, precursor drug is given that replenishes.
See, that was my point, that if you have a drug that instead of recycling of that dopamine or going absorbed back if it remains in the synaptic cleft for a longer period of time?
I think you are getting into a very molecular mechanism. Go ahead. Please answer. Please answer.
So actually with time it is not only the drug that is lesser in number, but the receptors that receive the drug also are less.
It's a degraded, it's disease that degrades, that over time you don't have the release, the recycling and all of that. So you're getting into very deep molecular, because these molecules and then subcellular organs that work at it that we don't have current capacity to target. But the drugs are like a high level lucky target that it turns out that that drug has certain temporary treatment. People have, for example, drug addiction, then it is basically because a molecule connects to a neuron and locks it in, and therefore you get addicted. Now if you can find another molecule that blocks it, you can probably treat drug addiction. So there are these what are called molecular techniques, but since this is a neurotechnology course, I'm not going to go into all of that and talk about devices and technologies to treat. Yes.
Probably technology called DBS, deep brain stimulation.
That is exactly all the stimulation we are talking about, the jargon, and you are correct. The jargon, the correct way to say it is called DBS, deep brain stimulation. So we have just your answer and all that we stimulate, and what we really mean by, because these regions of the brain that cause these tremor are deep inside the brain. So that's why it's called DBS, or deep brain stimulation, to treat Parkinson and potentially other things.
Risky and also very cost-effective.
I don't think it's any longer risky. So we have made a decision to latecomers will not be admitted. Okay, only single one-time excuse. Okay, so please come in, and we will not. That's it. Basically, one day that, next day you can go home. So I don't want disruption in the class. You take it seriously or don't come. I would rather have this class with 5, 10, 20 highly interested, engaged people than people who are coming late and not taking it seriously. Okay, so of course, like any brain surgery, any brain activity is risky, no question. It's an implant, but it's now become extremely reliable procedure. It is expensive, and there are other reasons. Yes, of course, anything brain, these kind of devices are high tech, it's expensive. It's also expensive because there's one company only makes it. There's a lot of monopoly. People will pay to treat, and therefore they make a lot of profit. So there are other factors at work too. It doesn't have to be that expensive, and it's no longer, because it's been tested out for a long period. So it's pretty.
But sir, if stimulation is associated, so I mean it can be done with wireless, because at the hippocampus region, if we can put a.
So it's not hippocampus. Hippocampus for memory. This is the basal ganglia. Yes, you. In theory, that's why we're getting digress. Let's talk over coffee. What he's talking about, like there may be other options. So when I get to the DBS devices, I'll discuss that. Maybe we can find a wireless technology and so on. And I don't want to cut it off, but at some point I have to respect time. So we are already a bit late. So we can have offline discussion on the topic. People who want to, somebody in your family has Parkinson, you want to talk about it, we can have early morning, late evening, those kind of discussion. What is the treatment for paralysis? If somebody got paralyzed, either, of course, it could be stroke or it could be spinal cord injury. So what can we do for a person who is paralyzed, let's say it doesn't have a leg movement?
Physiotherapy.
So you can start with physiotherapy, not so bad. Somebody else what spoke up and then didn't. Come on guys, I don't have enough chocolate to incentivize. Can I request, I give you that electric shock device. So if they don't answer, I give them electric shock. We'll try that approach if the chocolates don't work. Okay. What was that? Go ahead. Expand on it.
To stimulate certain part for paralysis, we can use the technique.
So what part?
Which is not working, maybe.
So tell me more. Maybe find a doctor to talk to. All right, let's come back. You and the medical person side by side, you two can talk about it and come up with a treatment. Yeah, but any mechanical engineer here? Okay, anyway, open your mind to all kind of. Yeah, exoskeleton. So you are medical oriented or engineer? And you're talking about this jargon like, aren't you supposed to talk about lumen and all that kind of stuff? And he's using the word exoskeleton. What's going on with the engineers here? A robotic exoskeleton. Well, we can think about. So there are, who knows what are the treatment. So, we are getting into, from medicine we are now shifting gears to variety of engineering, and so we will need a lot of engineering tools and technologies such as monitoring the brain. So, this is a cap where little white circles are the electrodes and the box is the amplifier for recording EEG. So, we are going to now, as I said, we are shifting from medicine to engineering, and we are going to need variety of engineering solutions for diagnosis and therapy and treatment, right. So, EEG, somebody out of this course, I am sure somebody is either making EEG electrodes, or you can be recording EEG and doing signal processing, or you can make these devices. And so, we are going to talk several types of, I am going to give you an overview of variety of devices. Imagine a picture of an operating room, or a, what do you see, what machines are implied, although because it is kind of medical, so a lot of these is a clutter and it's covered, but what is on the screen?
Radiological images.
So, they are radiological images, CT scan, MRI scan, bread and butter. You are not going to do brain surgery or anything like that without images. So, that is a part of neurotechnology. Unfortunately, I am not going to cover because I can't cover enough topics, but you should never overlook. It is a huge part of neurotechnology, which is variety of different brain imaging techniques. Brain monitoring, so the person could be on a computer screen and looking at brain waves or vital signs of the brain and so on. So, there are a lot of technologies that go into an operating room. So, engineers, people taking this course could be doing research. So, when I came to Johns Hopkins, as I said, I was doing a lot of work on heart at that time, and I ran into a physician, an anesthesiologist, who brought me this problem. He says he is in an operating room, and I mean so anesthesiologists are the one who make patients go to sleep, so to say, and the surgeons operate, right. But anesthesiologists also monitor the patient, all the vital signs continuously. You wanted to be a doctor, right? So, now this you are getting enough medicine out of it. I hope you do not regret the fact that you did not become a doctor, because now you can make a difference, which is, so I mean a person is anesthetized, so cannot respond and so on. A surgeon is operating, for example, on spinal cord or something, and one wrong cut, wrong move, compression, there is a lot of sometimes bleeding, you can press on a vessel, you have no way to know. Spinal cord, brain does not complain. The doctors in anesthesia, when during surgery, can monitor heart rate, blood pressure, the other vital signs to make sure that you are not dead, or the control of this. They monitor for enough so that you do not feel the pain and depth and all of that. But how do you know that the spinal cord was not intact? And it could take I mean seconds to minutes before permanent damage occurs. So, we talked about a technique called evoked potentials, evoked response. So, you stimulate the leg with the electrical current. So, if I stimulate my leg, the electrical current will go up the spinal cord all the way to the brain, and then I record the brain activity. So, every time I give a stimulus, I must get a response. If there is any break, spinal cord or somewhere, the signal should disappear, right, and you would get, the surgeon would get the indication. Now, it turns out the brain signals are so small that you can't, like, it shows up right away. So, you do a lot of what is called signal processing. You can process that in variety of signal processing techniques to enhance that signal. So, my first of this series of paper at Johns Hopkins were in this topic. You see, wonderful mix of medicine and engineering side by side. Those of you who are at IIT Kharagpur, you have an advantage. You are going to have a hospital, or you already have one. It is research oriented. This is where, and then doctor brings you a problem, engineers goes and solves it, vice versa. You have a new technology for imaging, you go and tell them, can you use it, and you can come up with a solution for intraoperative monitoring in this case, so the person does not get paralyzed. So, operating room can have a lot of techniques. One of the obvious one, even in passing what I said, is anesthesiologist monitors a patient who is anesthetized, which means is asleep or pain free or movement free. How do you monitor what is now called depth of anesthesia? How do you know that you are fully anesthetized? How, anesthesia, they give this different types of anesthetic gas. How do you know that you are anesthetized?
Doesn't respond to stimulus.
Sure. So you can pinch, doesn't respond, fair enough.
The brain waves,
What would they show?
I think they go into alpha. I'm not sure.
Repeated patterns.
Yeah. So you have both extremes. Obviously, perception. So the anesthesiologist talking to the patient, and before you know it, slowly the patient stops talking, and so there is no responsivity. So obviously, there are many things. You shouldn't be moving, right? You don't want to be starting to walk during surgery. So paralysis. You should not be able to remember anything. You should not feel any pain. So all of these can be observed, but still not very rigorous. So then you can revert to brain waves, and then brain waves shift into a certain frequency changes, and therefore from that you can calibrate depth of anesthesia. This was an invention by a biomedical engineer. So somebody like you in this class, given a problem, can come up with an algorithm of changes in brain wave. This person developed a technique called bispectrum. So because their changes are complex brain rhythm, and figure out what the depth of anesthesia is. You can monitor the brain directly on the brain. So this is what the brain surface looks like, and there are little, these yellow dots are electrodes on the brain. This is a study where patients who have epilepsy, intractable epilepsy as it's called, cannot be treated by drugs. Extreme. They get convulsions, seizures all the time. Then they have to, surgeons have to go in and remove the part of the brain. Remember that HM's example I mentioned? That is an extreme, and actually that's not no longer recommended or preferred, because they were trying. These days you just remove the cortical region that is mapped. So like that yellow area, maybe where seizure you've seen more. Those electrodes there are more seizure, they over here. Then doctors will now make a careful attempt to say, let's remove this part of the brain. What is the price to pay? They're removing some part of the brain. So some part of the brain function goes away. Well, brain is so amazing, and so much is not known, so much is complexly covered, so we don't know. But they try to protect certain things, like particularly speech. That's the one area they want to, because suddenly your humanity is lost. Loss of limb function versus no seizures. So you can live at least survivable life. Tough decision, right? Very tough decision to remove part of the brain, to lose something, but get out of the disorder. Same thing during tumor. They have to remove the brain tumor. How much to remove? If they remove a lot, you're taking a lot of brain. If you don't remove, some part of brain tumor cells remain, the tumor will come back. Very tough. Engineers have a huge role to play to figure out how to market it. In over Singapore, we are doing some of this. This project, epilepsy, is at Johns Hopkins. In tumor marker and imaging is a project in Singapore, where say, how do I help surgeons demarcate different regions of the tumor? So we use some nanoparticles to do the targeting, or looking at blood vessel change that are changed during tumor and so on, and then hopefully guide the surgeon to remove just the right amount of tumor. So many such roles for technologies. This is the deep brain stimulation kind of things, where but there are other things, right? So this also, by the way, gives you a bit of an architecture of the brain. Brain. You can see the little gray area is the gray matter, the white matter. There's a lot of fluids in the brain. It's called CSF, cerebrospinal fluid. Then in CSF, somebody gets meningitis. Its CSF gets infected, and then it can be very life threatening. Or head trauma, boom, head gets up, car accident, whatnot. Very likely you'll have this bleeding or fluid buildup, and then that creates a life-threatening situation. To detect that intracranial pressure, how do you detect pressure in the brain? How do you get heart blood pressure? Hey, just raise a hand. One of you speak up. Murmuring doesn't help. Not you, somebody else. Why are you also answering? You're answering somebody else. Blood pressure, come on, that's simple as that. So you also answered that. Somebody else. Come on, guys, why are you, some of you are so shy? So I was at IIT Gandhinagar last couple of days before coming here. There was a lot of discussion about education, and how to make better students, better future direction. And you know what was the number one thing that came up from various advisory people, research out there that needs improvement? What is that number one thing that will make students better, successful in their careers?
Participate more in the class discussions with the faculties. With the
You're giving complicated answer. One word.
Interaction.
Communication, right? So it's same thing, roughly what you mean. Can I have a raise of hands? How many of, and please, you must raise hand one of the two ways. Don't be passive, because I'm going to, so I made one rule, which is that if you come late, I won't let you in. It's very disruptive. If you don't interact and answer, I'm also going to kind of watch. Well, those who are completely non-interactive, I'm going to, at some point within a day or so, I know you're in the beginning a little bit shy, maybe you're nervous, you're stressful. You know, I understand that. I really understand. But come on, we're just friends here. I mean, you know, you're here to learn, so you should interact, okay? So if you are not continuously not interacting and participating, then in a day or two or three, I'll say don't attend. I don't want to do that. I want to actually give chocolates. I still have actually another bag. So please, new people, I want to have, anyone want to answer now? How do you measure blood pressure? Come on, it's the simplest thing in the world, right? You go to doctors, well, the first thing they do is blood pressure. One of you already gave an answer. Do I have to go and say you answered? That's all, you already answered, so you're a good guy. You over there in that middle row in the back, how do you measure blood pressure? You, yes, young lady, you in the back, how do you measure blood pressure? You are the, I'm looking at you who are turning face, you are avoiding. You have a choice, you can step out of the class or answer the question. How do you measure blood pressure? No? You don't know how to measure blood pressure? You've never had your blood pressure taken? Can you speak? Okay, are you afraid of me? No, I'm speaking to her, the young lady at the back in the middle, right? So blood pressure. How do you measure heart rate? Okay, all right. I don't want to pressure you, but please relax, participate. All right, let's come back to that in a minute, but somebody else wants to answer because I'm trying to work out. You've also answered. I want somebody new. How do you measure heart rate or blood pressure? Yes?
sphygmomanometer.
Yeah, so sphygmomanometer, just, you know, you're following these guys. And like, can you bring it to like nice, simple English? I don't know Bengali, Hindi will do, but simple language. What is it? How does it do it? It's a barometer. Do you know what a barometer is? Okay, all right. So it's a blood pressure size. How does it do it? What is it? What does it do?
So basically, how barometer works, it has a pressure-sensitive fluid. So when the pressure rises,
okay, good. All right. So the idea is that there is a pressure in the air. There is a pressure outside. There is a pressure inside. So if you can compare the two, so the first of the thing is like this mercury kind of thing where you puff up, you put a little cuff on the arm and pressurize it, and when it stops the blood flow, the outside and inside pressures are matched up, and that pressure is now seen by amount of fluid rising or mercury rising in a tube, and that's one way. Nowadays, of course, these are electronic. You can put a pressure sensor inside the artery. So you can put a needle and put a pressure sensor inside. Okay. How do you measure pressure in the brain? So what happens is, you know, this is a huge problem in India, but many places. Car accidents, sports injury, head hits, there's a damage, there's a blood vessel pops up, there's a fluid builds up, pressure goes up in the brain, you could die. How do you measure intracranial blood pressure? Okay, so it is today's homework, research homework. I'm not going to grade it. It's curiosity driven research homework to research. It is not a entirely solved problem, although there are a lot of technologies out there. So intracranial pressure is a big thing, but you can see the picture because you have a skull. So how do you, I mean, You know, so let's see if you can come up with a good answer. Yeah.
By intracranial pressure, is it like blood pressure specifically, or is the fluid?
Fluid pressure, fluid pressure. Good question. So I started because I didn't expect, so I backed up into blood pressure as a way to everyone to understand what is pressure. But what we really want to measure is pressure inside the skull. And if it goes up, then that pressure will squeeze on the brain, and a person can die with something called herniation, right? So measure, how to measure blood pressure? Somebody, just for the sake of complaining, how do you measure heart rate? Come on, somebody new. Okay, I mean, somebody who has a very, not you. Come on, why? So going back to the communication, it's by far going to make a difference in your life and career as a professional, by far. So today's your inability to interact with nothing at stake, right? 5 days I'm going to go away. All the people in this room will be gone. Why are you shy or afraid? It's going to hold you back, right? So please relax, communicate. I mean, what's wrong? Several of you have given right and wrong answers, and it's okay. All right. So all of you, those who are not participating, get to know. So Government of India, Prime Minister actually specifically told me, he came specially to Singapore to tell me that make these Indian students be more dynamic and active. No, he didn't say that, but I'm saying that I think my here is not to give you, actually a lot of things I'm presenting to you, you can look it up on the internet. But things I'm making you do to think and dynamically interact, I don't think you're going to get on interact. So please interact, please participate. Yes, ask questions, that's even better.
Is there like measuring blood pressure? It's a very non-invasive technique, and but if
It is non-invasive because it's better that it's non-invasive, but non-invasive is not accurate. So
like everything happens outside, we don't need to poke anything. Like for example, if we have to measure some intracranial pressure also, like we need to insert, like we need to drill a hole or something.
that's why it's a homework that I do. You do some research. Yeah, think more about it. Yes, you can drill, and that, is that a good or bad? So tomorrow we will be looking for the best answer. I mean, this is the world's best chocolate waiting for the person with the best answer. No, I'm just in interest of time, I'm giving it as homework. I'm glad that some of you have other questions or curiosity. Should I drill a hole? Should I not? Is there another way? What is the best way? As I said, it's not a perfectly solved problem. In fact, our own blood pressure, the sphygmomanometer, all of that, it's not terribly accurate. You'd rather put a blood pressure transducer inside an artery, but that's too risky. So you don't want to do that. So you do what is called non-invasive, preferably. So there are many ways to develop these kind of solutions. Please research it, and we'll come back to that. Okay. Okay. So this is what an implantable. Why am I? Okay. So it looks like they want me more than the screen. How come I'm not shifting to the screen? Oh, okay. All right. So this is what a implantable deep brain stimulator or pacemaker would look like. It's in a titanium can with a little wire going out, and that wire, then in this case it turns out to be, I think it's for pacemaker, but something very similar for deep brain stimulation that goes deep inside the brain. So engineers will need to build this kind of implantable devices. It's in titanium can to perfectly protect it, seal it, and totally save it, so the electronics can save for many years. This is what devices look like. But as I said, something simple. You can detect pulse, you can look at muscle signals, you can look at oxygenation and the fingertip, the finger oxygen. So what happens if somebody goes into accident, there's some bleeding, body to preserve, what would it try to protect more? Vital organs, but of which is the most vital? Heart, brain. So what happens is the blood essentially reduces from the periphery. So those of us, not all of you, you won't relate to what I'm saying. For those of you who live in very cold climate, that's what happens too, that all the blood goes inside the body. So our fingers feel really cold. If you go to Kashmir or something, you might see it, because peripheral circulation reduced. So measuring oxygen and flow at the fingertip, it gives you a hint about the brain, something is wrong with the brain. So, that getting up oxygen or at the fingertip is a very, very indirect way of seeing whether everything is all right, but it is very easy to do, though sometimes it has that advantage. So, there are a lot of these solutions. So, you see there is a panorama of technologies. I haven't even covered everything, just a good sample. Sorry. They are used in all kind of situations, in operating room, in emergency, like an ambulance comes, somebody is accidents, operating room, in military. There is a need for neurotechnology for like this kind of diagnosis. So, question: how would you monitor? Sorry, my grammar could be improved. A person, or know that a person is fully anesthetized. So, right, we discussed that. How would you know if somebody is dreaming? Come on. That's not how we work. Oh, yeah. See, new person.
The eyelids move even when eyes are closed, and the breathing is related, and the heart rate is related. So, based on that.
Okay. Anyone else wants to complement to that? EEG wave. Okay. Right. So,
one more thing can be done.
Okay.
Very easily, simple. You come towards him and move away, and he's still seeing there. He's in state of loss, not focusing that right.
I have not heard about it, but we can think about it. You can bring me a research paper in New England Journal, then I'll believe that. Yeah. So, look, I think this was meant to be an open-ended provocative question. Frankly, I don't think we know. So, what you are describing is what is a part of a sleep stage called REM sleep, rapid eye movement sleep. So, we think that a person is dreaming, but we do not have a way to know. We do not even know how. Do we know why we sleep? Anyone know why we sleep? So, the way I paused it, you are pausing and so on. Yeah, we do not know, really we do not know. So, 1/3 of our life we have no idea why we do that. I mean, there are certain obvious benefits, resting and all that nonsense, but we really do not know. So, same thing with dream, Freud probably knew, but it is not really easily understood. But yes, eyes are moving, something is happening, brain wave is changing, so we may think it is dreaming. You can do brain images and maybe it will show some activity, but it is one of those open-ended questions. So, I raise these kind of questions not for expecting a perfect answer, but how would you sense some vital information of the brain from heart signals? Anyone can say that? I am looking at heart signal like heart rate, heartbeats. Can you tell me something about the brain? That's worth a chocolate, I would think, although I am going to run out of it. So, I will break.
For example, like if you have arrhythmia or some irregular heartbeat or something like that, you can know what specific part of the brain, like cerebellum.
Yeah, I don't think. Oh, well, cerebellum is the back part of the brain, which is for balance and movement. So, not. The arrhythmias are abnormality of the heart. So, that is not the correct answer. We can try for a minute, or I think you might want to do some research on it. Unless somebody has, yeah, go ahead.
I'm not sure, but like suppose if you have some brain disorder, and suppose you're paralyzed and you want to move your hand, but your hand's not moving, so maybe your heartbeat increases.
Yeah, I get. Yeah.
Because if the intracranial pressure is high, the heart rate will be low.
Yeah. This guy is good. Okay. So, sorry, you can pick it up later. Well, that's a really great answer. So, when the intracranial pressure goes up, then it presses on the brain stem and the core regions of the brain, and that affects heart or breathing or respiration. Remember a few minutes ago I said that can be almost life threatening, these kind of things. So, a heart rate change could be very indicative of it. So, professor, for you, yesterday you were measuring heart. In my opinion, you need to measure heart rate variability. The heart rhythm itself means nothing, because all of you will sit here with different rhythms. And yes, of course, meditation will lower the heart rate. And maybe it is good, but I am not sure that there is necessarily a causal or proven thing that somehow lower heart rate is better. I think it is better, at least have a lower heart rate. That is generally good, physically fit people. But heart rate variation, See, we are all able to react our heart rate to stress, to running, sleeping, exercise. If you cannot do that, something is wrong, and that is controlled by brainstem and cortical rhythms. So heart rate or heart rate variability is in a way an indirect indicator of brain function. My point to you is a lot of medicine and diagnosis is indirect. Please.
Then this can be one of the answers for the homework question.
Which one? Which question?
The measuring the heart rate.
Yeah. I mean, I don't know which particular question are you referring to.
Intracranial pressure.
I don't think so, because that situation is an extreme situation. At the very end, the pressure is so great that your brain will just collapse because it's impressing on it. It is okay.
That we need to derive a function between the both.
Yeah. See, did I say you're wrong? I never said that, right? I think you made a very. See, this is one of those things I want to tell you all. Why you all need to participate in, because they are not always. A lot of these things are yes and no answers. Some things may be. A lot of it is open for discussion, and your engaging in like this will allow me to teach you in a more enjoyable way, because we can understand and learn to think pros and cons. So point here is intracranial pressure is going to go up. It's compressing. So certainly it's affecting heart rate. Now one of you asked a question, do I have to drill a hole in the brain? How many want to get your drill a hole in your brain? Please raise hands. So if you come up with this kind of indirect measure, even if not perfect, that's a start, right? It's a start. These docs will like it, unless of course they are neurosurgeon, then they are they don't make money, so they say no, drill a hole. But otherwise they like this thing. So it's a reasonable answer. Is it an accurate answer? Is it a solution to, I don't know. So that is why it's provocative. So some of these questions I'm going to give you, this could very well be a PhD thesis with still not a perfect answer. So it's plausible, and it's one of the non-invasive ways you put it, as opposed to drilling a hole and putting a pressure transducer, invasive way, and then all kind of alternative creative solutions to these. Okay, so it's great. I'm glad you guys are thinking like this. Can you read mine? Yes.
Sir, is the intracranial fluid transparent or translucent?
I think you two get together and you talk this gibberish, intracranial translucent lumen sphygmomanometer. Okay, otherwise you can come back to simple. I'm relaxed. Think about it, right? Let's in the interest of time, let's move forward. Please research that a little bit. Don't speculate. So it's not yet ready, but research it a little bit. There is possibly, because right, you're almost what I think intracranial pressure should affect the cerebrospinal fluid, and therefore there should be a way to measure it, but you need to research that a little bit. So you're on the right track. It's invasive, so it's something to the. Okay, let's go on to another question. Can you read mind? Can you know what I'm thinking? So when I was young, there was a song that I remember. Sorry, it's going to be in Hindi. Sabd kho ke ek dawai, kyun na ajmaye, right? You may remember, right? So EEG is like sabd kho ke ek dawai, no matter what, so let's do EEG. Somehow we'll figure it out. Okay. Anyone else? Can we read mind? Sorry? Psychologists read mind. Maybe your colleague should come to the class and ask her whether. Can I suggest this? I have a feeling, I don't know what time we are in. All of you should get together and argue, fight with each other about the right answer or tea or coffee. Is it a good time for tea or coffee? Because can you read the mind? And of course, you all have many interesting answers. Can I just flash a bunch of research questions? Research or download, you can write it down if you like, but I mean, this is purely voluntary, but to raise your curiosity, he has got the right answer, so Sudeep has the right answer, take a picture for God's sake. Today, why even write down and take a photograph? But these are what's asking, right? Which part of the brain is involved, what are the structures? I mentioned hippocampus, we look at cortex, I mentioned brainstem, basal ganglia. There are so many different parts of the brain that you should get to know. neural organization of the cortex, I showed the picture, layers, layer 1, 2, 3, 4, 5, 6, what do they do? Spinal cord seems, we saw that, but what is happening in cross section and what's where? Nerves, whoa, that will be very, I showed one picture, but that was like a cartoon, it was not real. So what are the nervous system? Did we miss some other neurological disorders? I covered a few of them. And then I already mentioned that, right, like I didn't really cover brain imaging technology, I just mentioned CT and MRI as we went along. So there are a lot of things for you all to know about, okay? And so I think that this is end of lecture 1. So I had wanted to cover 2 lectures during that time. Yeah, that's okay. I think we are done. Oh, you want that slide back? What is this? I know, but see, my screen has gone blank. It's got black. I don't know why this keeps up. I understand that my screen here, there's a laptop issue. I know.
They are taking some. Okay. Suman, will you please just look, give the display of the.
Yeah, it's okay. It just somehow sometimes this is happening. So why don't we go back? So apparently, I think the PowerPoint crashed. It's telling that even the laptop needs coffee. I don't know. See the file. Show Windows. So lecture 1. Oh, it's in the right side. You're right. Okay. So let's go back. Bring it here. Here. Oh no, this is something else. Lecture 4. All right. Let's take a break. It's okay. That's not really. Ask your neighbor. So the point here is, see, please, all of you, right? First of all, if you want to answer, did you enjoy the first lecture? Raise hands, please. Oh, 100%. You'll all get an A grade. You didn't raise it, so because you came very late. So we are not going to come late. If I come late, that's okay, but you guys cannot come late. Let's make the most of it. If you did enjoy it, that if you did really mean it, and because it was very disruptive, we lost at least half an hour on all of that. So I'm hoping that we can have more enjoyable and interactive. I did go a little bit slow to raise the ideas and discussion and create the spirit. Please participate. It's good for you. Okay, philosophically speaking, I'd ask, like, you guys were very good about answering the question, did you enjoy the class? So I'm going to ask this question. It has 2 parts. How many of you want to get rich? Raise hands. Okay. How many of you want to get famous? Raise hands. Okay, 50-50, sort of. See, there are 2 things to do in life, get rich or get famous. Either way, you're going to need to communicate. If I was giving a dull, boring, stupid lecture, you'd all be asleep. Wouldn't be fun, right? So I learned communication. You go, graduate from IIT or wherever, if you want to communicate, you're not going to get a, first of all, job in an interview, and you're not going to get promoted, and learning to be nice to the boss and friendly and smart and happy. You sit down in a little group in a team, those who interact are going to do better. So going back to IIT Gandhinagar, one of the advisory senior, the person that, this was, you can always mention Harvard and it sounds good. And they asked alumni of Harvard, what did Harvard teach you that you valued most? What was the answer? They didn't go there for physics, they didn't go for math, they went for communication. There is a cultural aspect of it. Indian culture taught to be quiet, Chinese culture is like that, obedient, don't speak too much, all that. There's a gender type, women seem not to be encouraged or socially or something, it's holding you back. So please, this is a no cost way to be happy, friendly, because at least not till tomorrow we are not giving electric shock. Tomorrow we might resort to that, right? So no penalties. Do you have any other, is there a IIT, do they allow caning or something like chabuk maro, I said nothing like that, right? Okay, so we're going to try chocolatey route, right, as far as possible. So what's the harm? Money, I don't know, that's an option. Just enjoy, interact with each other so that you will can we read mind? You can have some argument. Please don't fight on that topic. It was wonderful to see yesterday about meditation. All of a sudden, a lot of you woke up because people in Asia seem to want to know about that and have an opinion, and somebody asked a very intelligent question about different types of meditation and so on. And I told this, one of my former students who is now in China, he sent his students all the way to Tibet to study EEG, doing different kind of meditation for different monks. So, you know, so much fun we can have interacting, discussing topics. At the same time, you know, we are here to learn, so let's be rigorous. Okay, so great. Let's take. It's 11 exactly 11:01, so we come back here at 11:15. Correct?
11:15 is right.
Okay, and please assemble it in so that we don't waste a lot of time coming in and noise and shuffling. Okay, thank you all.
Just I want to give an announcement. On Friday we have some quiz test, right? And
Okay. So, thank you, sir. Oh, yeah. Thank you for. I'll remember next time. So, as I said, you know, it's a lot is learned through these discussion and interactions because you know that's going to make you curious and come up with ideas and answers. The factual part of brain, 80 billion brain cells and a complex network, and it's just going to be too much, right? So you can only remember, but at some point something will create your interest in a topic, and that's what I'm here to do. So please interact with each other as whenever you get time with me. As I said, early morning, late evening, we can, time permitting, we can have discussion about certain topics. But at some level we should also be very objective and scientists and engineers, because some questions, is there God, is why is sleep, they get very philosophical and deep. This is the course that cannot cover that, but we can be more rigorous about brain technology, clinical applications, and that's what we are here for. Okay, but I will give you always some provocative questions, and I really value and enjoy your participation. Okay, so now we're going to switch gears. What I want to do is to now give you a rapid overview of the research cutting edge, right? So morning was tutorial, more biotechnology oriented. Now we'll jump high level to give a rapid overview of the technology. It's pretty much more or less mapped on what I do in Singapore. It's, you know, I direct a brain institute, as I told you, Singapore Institute of Neurotechnology, and we do research in many frontiers. And of course, that's being done either by some of my faculty or postdoc or students in many of these topics, but it will give you a high level overview, not entire field of neuroscience or neurotechnology, but many interesting topics. Okay? So this is where we're going. So this is like an overview. So so this is in two parts. Sorry, I realized that I had lecture 2 and I was delayed. So let me do the overview first and then go deep and advanced later. Okay, so it will be in 2 parts. So let's do the overview of some of the frontiers of neurotechnology. So just as a kind of a background, right, as I said, I started my work in heart and heart monitoring from the pacemakers and defibrillator, and that has revolutionized cardiac care. It's really one of the last 20-30 years of amazing success stories. People live long because heart disease can be treated. There are drugs for it. There are stent kind of devices. There are pacemakers, defibrillator. So the point is there revolution applicable elsewhere? This is a little device. It's called a pill called Bion. It's actually an electronic stimulator for muscle. So it can be implanted to stimulate the muscle to restore muscle function for people who might be paralyzed and so on. So there is some interesting technological evolution. So what are the frontiers of neurotechnology? Some technologies that have emerged. Well, one of them is cochlear implant, right? So a cochlear implant is one of those tremendous success stories. A stimulating like device essentially is able to restore hearing. And what's the basic idea? That you can pick up some sound picked up by microphone, as you can see this point by point. Where is my cursor? Yeah. So you can pick up sound from a microphone like in a ear. Sound is processed by a signal processor, and then it analyzes speech and it digitizes it into frequencies. In different frequencies, if they can be decomposed, then they can be used to stimulate the nerves in the or what's called cochlea in the ear. So as a device, of course, it does this coding. Now, of course, this kind of microphone, all that is outside, and then there is an implantable part of the device that's here. This is the implantable part on the left side, and this is the external part on the right side. And then transmitter sends the data, the implant goes into the brain and so on, and then it stimulates. Actually, to be honest, to say stimulates the cochlea. Cochlea is like a spiral-like structure in the ear, inner ear, where nerve endings come in, auditory nerve endings, and they are what's called hair cells. So this will stimulate that cochlea to provide electrical stimulation to that as a alternative to natural stimulation, right? Natural stimulation is from sound and sound vibration, which get converted to electrical message of the nerve to the brain. As opposed to that, what we are doing is providing an electrical signal, so that that electrical signal stimulating will give a perception of sound. And particularly young people can pretty much learn to have a natural dialogue, because their brain adapts to this unnatural electrical stimuli. Terrific success story. It works. Of course, it's implantable, it's expensive, it's not the same as hearing aid, but when there is this kind of a problem of the ear, cochlear implant would be very useful. And and it embodies fantastic electronics, miniaturization, excellent success story. Emerging success story is a retinal implant. A retinal implant or retinal process is the same way to give vision. So now a retina, which is a part of the eye, which has these cells called rods and cones. Rods are cells that sense kind of black and white and intensity. Cones are color sensitive cells, and that's in a structure, retina, inside the eye bulb, right? So if you look at the eye bulb, somewhere in the back is the retina, and all those red rods and cones are in there. So if I see some picture, it's going to go through the lens and then into the eye and into the retina, and then an electrode array, this kind of an electrode array, 10 by 10, 30 by 30 tiny little dots, will stimulate and create a perception of sound. These are called phosphenes. Can you wait outside? Since you're walking in rather late, so it disturbs us. So just stay outside and I'll call you in later. Try to come on time next time. Okay, come on in. Find a seat somewhere. I don't know.
Here you can come.
Yeah, there's one here up front, or yeah, up here. Okay, so a retinal prosthesis is for stimulation of the retina to give a perception of light, and there's something called phosphines. These are artificial sensation of light that you can see, and a person kind of at 30 by 30 can tell that there is a person there or a door there or something, but cannot do much more than that. But it's a very vibrant, evolving technology, and there is now some commercial product, very expensive, very limited, but it's happening. So nice evolution. Of course, the deep brain stimulation, we talked about it, right? So pacemaker like this implantable device, and then it goes into the brain, and then you have these electrode that is, you see how look at this surgery. So these pacemaker like devices here is threaded through the neck all the way into the scalp, and then into there. You want to avoid drilling holes in the brain and so on. So it's, and wires running outside and all that. So this kind of a device, and then you can see it's deep brains, this area where this sort of deep brain area where basal ganglia, where motor control is, and by stimulating. But essentially it's like a pacemaker like device. The difference in a pacemaker is that if you give electrical pulse every heartbeat, or if you miss a heartbeat, you give a pulse. In this kind of deep brain stimulation, you give high frequency stimulation. Doctors, inventors empirically found that high frequency stimulation suppresses tremor, so oscillation. So it's very successful solution. This kind of deep brain stimulation technology is now being prevalent in many other diseases, epilepsy, depression. So it's the frontier. It's opening up new interesting research areas and technologies. A little bit more about deep brain stimulation, because so important that you can see the pacemaker like device. This is the lead, and the lead you can see all these electrodes. That's where you apply that electrical current, and then the stimulation is high frequency stimulation is given. So here is like, I'll play a video. So this person, stimulator is on. So stimulator is turned on externally, and we had talked about earlier, like wireless communication, so that the implantable device wirelessly gets the information. Stimulator is off, you see he has tremors. His hand is oscillating and it's not stable. As soon as, and when the stimulator is off, there is no effect. As soon as you see he cannot hold this, as stimulator is on, tremor go away. Remarkable, right? It's such a miraculous solution. Not so easily understood mechanistically. There's a lot of research still to be done, and in fact ongoing, but it's powerful demonstration, and it does also affect, I just showed the video of the arm, but also of the leg and rigidity and some effects like that. So my last line there is, what other disorders of the brain do you know of that can be treated that way? And there is a high level of emerging research in this area, and maybe you can do some research and find out for yourself. I gave a couple of examples like epilepsy and depression. People who are depressed, of course they first go with medication, but these kind of stimulator, in this case stimulating some nerves, has some therapeutic effect.
Sir, anything has seen in West syndrome?
In what?
Any kind of work been seen in West syndrome?
West syndrome? I don't know what that is.
It's an epilepsy.
Okay, sorry. So so there are certain, so I didn't know the jargon, sorry. So yes, the epileptic seizures, vagus is a nerve. For some kind of epilepsies, vagal stimulator is shown to suppress epilepsy. So it's an option. There is a company that makes direct cortical stimulation, so similar device, but put it on the brain and it stimulates. Again, it's perhaps with vagus it's easier because the nerve you can stimulate at the neck level or something, but for direct cortical stimulation is brain surgery, so it's a little more extreme, and much more extreme is the surgery to remove the brain.
Just a simple question. In stimulation, it is just the excitation, or does it show any kind of this neurotransmission and all phenomena? Only excitation, it's simply excitation.
Yeah, so this takes a much deeper discussion because there's a lot of science and engineering behind it. Most people will think of electrical stimulation as excitation, but it can also suppress or depress. It can also interrupt communication. Consider this, right, that brain cells, 10 micron size cells, are packed in 3 dimensions with complex network that I showed you. How are you going to figure out what is exciting, what is inhibiting, what is suppressing, and all of that? So one issue here is matter of excitation versus suppression or depression. The second issue is that when you apply electrical stimulation, reason why I do it, because we can control it with a battery, electric circuit, we can do it. What does it do? Does it directly either stimulate or depress a brain cell or network, or does it have an indirect effect? So by doing that, it alters the release of neurotransmitters or other chemicals. Both are possible. Each situation is unique. Presumably epilepsy is a disorder of electrical chaos in the brain, and so it's generally seen as very electronic, and so in that sense it is potentially disrupting this oscillation. Versus Parkinson is occurring because of dopamine deficit. So electrical stimulation is altering the circuit that was altered because of loss of dopamine. It's not producing new dopamine, it's not. So generally these, currently our solution is pretty much electronic and electrical, either excitation or depression, but probably have secondary effect on chemicals and all of that, but we can't control them so well. Okay?
So this neurostimulator for Parkinson's patient, do it need to be optimized for every patient, or?
Yeah, it is. So the question is, do these devices, do they have to be optimized? Yes. So that's why they are programmable. As for the matter, even pacemakers and all of that, they are optimized for each patient, but it's optimization is a little, again, very elaborate. These are each thing you have to have a long answer, so I cannot do that because of time. But you know, generally in that frequency in around 100 to 150 hertz is effective. How much current to apply is optimized. You know, how often do you turn on? Because if you turn on a lot, you're going to drain out the battery, then you'll need a new implant. So you have to do it selectively, so that's optimized. Where that implantation, it's such a small area, about 4 millimeter area of the brain. So it's very precise positioning that has to be optimized. Yeah, so it's case by case. There are similarly implantable spinal cord stimulators, you know, because earlier in the day we talked about spinal cord stimulation as a way to treat paralysis and so on. I mean, in the sense that it can effectively initiate electrical stimulation that can then go to the muscle, and hopefully there is some restoration. So there is some advanced research in that direction. The other area of spinal cord stimulation very interesting is for pain. Pain is one of those really worst things. You know, people cannot tolerate pain. You know, people who get cancer, certain type of nerve injuries, you may know people citing, you know, the pain is a, you know, humans cannot handle that, and suppression of pain is a big deal. And so of course there are drugs and they are narcotics, so they are addictive and they are all these problems. So there are devices that are, by stimulating the spinal cord, can block. The conceptual theory is that it blocks the spread of pain perception, you know, as if like I have a feeling of pain in my lower back or my leg, and I stimulate the spinal cord, somehow I am blocking the message going to the brain. You know, sort of. I mean, it's not a very accurate answer, but these kind of stimulators do work and are popular. Implantable, more specific, but require surgery. You may prefer non-implantable way of providing stimulation. They are not so specific because you don't directly target, but you know, cheaper, simpler. There are emerging hot technologies like robotic brain surgery. You know, after all, in brain you must go very precisely somewhere and remove the tumor, or place this micro electrode for deep brain stimulation in a very precise manner. Every piece of brain or whatever it's removed is high risk. In medical training, neurosurgery has the longest residency period. You know, you do this and surgery, and you know, because of you have to learn this complexity in 3 dimension and continually improve the skills, you may not get in a practice. So anyway, the point is that brain surgery is a very elaborate area, and there is a tremendous role of imaging and robotics and so on in this field. So that's going to be very emerging frontier. Okay, so questions, right? So I should all be able to slowly answer. What causes Parkinson's disorder? Okay, very good. Everybody knows. What are other neurotransmitters? Yeah. Yes, very good. I'm glad some of you came well prepared, interested, educated. I see still some of you refuse to participate, refuse to get interested. So please try. Yeah, so good. And you know, try looking up. Nowadays everyone has phone, you can search these things. So yes, there are, you know, just because I mentioned dopamine, but there are many different types. There is what are called excitatory neurotransmitters, and there are inhibitory ones, right? And serotonin is kind of one of those feel-good neurotransmitter, makes you, you know, and so there is a lot of role. Acetylcholine, these are chemicals that connect between, you know, synapses or neuromuscular junctions. Many of them, like and so on, were affiliated with Nobel Prize and such. So brain is both electrical and chemical and many other things, genetics and whatnot. So you can always relate to these. What is the how is the sound converted into the stimulus for a cochlear implant? So microphone. Transmit. I always like if new people are so. Then what happens? Go ahead.
It transmits to brain.
Not brain. No, transmits to where?
Electrical signals.
Yes, to which organ?
Cochlea.
To cochlea, right? So cochlea, like a spiral-like organ, which has a lot of fluid in it, but also a perfectly arranged set of nerve fibers through that spiral, from very high to very low frequency. So you put electrode wrapping that. So if you stimulate here, you get high frequency. If you stimulate here, you get low frequency, and the whole spectrum. Okay, and so that is how the cochlear implant would work, or at least in the perception of sound. Brain figures it out. We don't know, but somehow these frequencies assemble into speech. Yes, hello, high frequency, hello, low frequency, right? That's fine. But why is that a speech? So speech people decompose in what's called phonemes. That's our way of doing it. Brain doesn't know if there is a frequency or phoneme. Right? We don't know what the brain's code is. You know, you need to understand. We don't know that. But still, somehow brain learns. Broken up frequencies, because cochlea is arranged that way. So by having enough, typically about 16 electrodes will reconstruct speech. And particularly like young people who are more adaptive, their still brain is plastic, learning, will pick up, convert that into perception of sound. Can they hear music the same way we can? We don't know. But it's very, very interesting. What does a retinal process stimulate to produce vision? What does it stimulate? Retina. Okay, but it's, by the way, not the only way to go. You can stimulate a retinal nerve. You can directly stimulate the cortex. So, and in fact, even in retina, what's called epiretinal, which is on retinal surface, and subretinal, below the retina. So it has to do with how retina is organized. So retina is organized by these rods and cones and so on. So actually, it's kind of reversed. The retinal ganglion cells on the top, and then rods and cones, and then there's a layer of nerve cells and so on, and it's multi-layer preparation. So you can put electrodes below or above. You know, all these different approaches are being tried. Generally, that's the currently prevailing solution. Futuristic, and actually the very first of the vision prosthesis was cortical, but it kind of went out of favor because, again, who wants brain surgery? It's too much. And also, by the time when you go to the brain, this nice organization, like cochlea has a nice organization in brain, then it's lost. For speech, it's totally lost. We don't know how the language is organized. But for vision, there is some organization, but not so good. So when you are at the cochlea or at the retina, in a retina, there is a nice 2x2 organization, right? So you can, when you stimulate, you get phosphines to produce the picture like a camera would. But once you go into retinal nerve, or in cortex more complex, but by the same token it might elicit more higher level perceptions like movement. What does a movement mean, right? Why do you see my hand moving? That's a higher level person. So maybe those are the kind of things. There's Hubel and Wiesel got Nobel Prize by looking at neurons which only reacted to motion. So, you know, cortex, yes, please.
Regarding emotion, can we implement something by which you can control the emotion?
Oh, hold on. I was saying motion. I don't want to go to E. E letter, you're scaring me. So that comes later. That's, you know, when I become a Maharishi, I'll deal with emotion. At the moment, I'm just not even a Rishi. I'm one level below. So motion, so I was referring to motion. The Hubel and Wiesel's work was that they did study in cats where they put microelectrodes and they showed a picture to the cat. And some neurons would be active only when I go left, and some neurons active when I go right, or they see an edge up or down. So they proved that brain and its neurons in this 80 billion organization. Oh, first time we now realize the brain has organization, and within organization there's microorganizations like certain visual structures, and that there are specializations like neurons that for motion. Yes.
So its brain is basically organized hierarchically, like level of fineness it's seeing.
It's, you know, again we are going to get into these. So sorry, once in a while when these questions become very deep, I'm going to avoid it because of lack of time, and you know we'll get digress from where we are trying to go. So entire structure of brain and organization, how am I going to cover it in few minutes? Or topic like emotion. I mean, these are much more elaborate, complex topics. Occasionally, of course, I don't have the expertise, so I can't answer those questions. I'll just be speculating. But yeah, I mean, you know, we are human beings. We like to look for structure, right? We like to say, how is society organized? Oh, we like that we have a prime minister, and then we have ministers, and then we have secretaries, and so on. We like organization. Really, I mean, it's fine. So it's one way to look at it. The brain is hierarchically organized. Brain isn't. Those are brain cells. See, the utter complexity is, these are 100 billion cells with 10 to the power 15 synapses. What do you mean by organization? It's what we are trying to bring some logic, because we can't take care of 100 billion. We can't, you know, they have 1 billion Indians. So we can't deal with it. So we organize, right? We organize in states, we organize along language, but it's our way of structuring it. So same thing with brain. We look at a picture, and anatomists will organize it by different shapes. Hippocampus has a certain nice shape, so call it hippocampus. Physiologists will organize it differently, right? Layer 1, 2, 3, 4, 5, certain types of neurons, and micro people will go there. So yes, there are many types of organization to make sense to this complexity, right? How is universe organized? You know, I mean, you know, it's organized by galaxies and stars and planets, but we don't know how black energy or black holes are organized. So yes, organization is there as a way to understand what we see. Okay. Coming back to emotions and so on, such a higher order, it's very hard to. So in brain, we call it structure and function. We see some structure and we see some function. So moving my arm seems like something we beginning to understand. There's a structure, motor cortex, which leads to function. Emotion, memory, learning, these are highly distributed, not easy. However, the crazy part of it is that once in a while something like a stimulation can, like a emotion, a depression is a kind of an emotion, and you know, a stimulation of some area reduces feeling of depression. It's like a black magic. I mean, it's kind of scientific magic. You know, sometimes it's a little bit of serendipity, it works, that it could affect, but we don't fully understand. So one of the things in a course like this, we must be rational, right? And we leave speculation at what time of the day? 8 p.m. Morning, a little bit, we have brightened shape, we have cutting edge. I read in this web that there was this, and what do you think? Then we deep questions, we leave it for late nights. Okay. So you know about retina and all of that, okay. All right, let's try a quiz question. Take a look and let's see. Oh, sorry, the last got kind of mangled up, so please ignore this whole research download. It kind of somehow editing. Okay, how many for A? Okay, how many for B? How many for C? So it should add up to 50. If it doesn't add up to 50, we'll adjourn the class, because we don't want people in the class who have no opinion, no idea, no decision, no conclusion. Shall I try again? Please, I am not keeping track. It doesn't matter, but please select, okay? Otherwise, I'll have to get into all the formal testing and all that. So please, how many for A? You know, none. Okay, B, none. C, I want to have a count. Oh, so all of a sudden, suddenly everybody woke up. Okay, all right, some decent number. How many for D? Some smaller decent number. How many for E? Okay, so the answer is D. The reason is that you're not actually rods and cones are actually the receptors that look for light, but they are probably the ones that are gone or damaged. So it's the cells below that, the processing cells, that are between them and the retinal nerve to carry the message. So electrical stimulation is going there. Certainly it's not an artificial eye, and you saw the other things, okay? And so you see, kind of, it's a little bit more advanced question if you're really listening to what I was saying.
Sir,
yeah. Yeah.
From what I know, the rods are responsible for luminance and the cones are responsible for chrominance. Luma and chroma, they both contribute to the picture.
Yes.
Now we are getting the picture.
Yes.
So why not that is the?
But because the reason why you have retinal process is because those cells are gone, they are dead.
Oh.
So then what are you going to do? They are themselves gone. Then if they had it, you didn't need it.
Okay.
Yeah. Okay. All right. So you're right. They have a luminance, chrominance. Again, I think these people have these. This front row people is bit problematic. Okay, I'm going to make you sit down. They use these words like luminance and what is that, lumen and all that nonsense. You know, keep it simple, guys. You know. Okay, no, you're right. So luminance, chrominance, or black and white and color.
Sir, in deep brain stimulation or other implants like cochlear or retina, is there fatigue cognitive?
Oh, definitely. Yeah, absolutely. See, so these are a lot of deeper questions, because what happens is there are a lot of issues. For example, retinal processes, so I'll come to that, but let me ask a question, right? So I said, so in our retina, we have about 250 million rods and cones and all that. I don't know the exact division, but something of that level. Current generation of retinal processes is 30 by 30 pixel, although really that has just been approved by the 10 by 10. So at 30 by 30, I can kind of vaguely say, maybe you can tell a face, or if in a room you can see if the door that's dark or white, you can do that. Some people learn and do better. Why are we limited to 30 by 30? What do you think? Why aren't we making million there? You know, so you can add a lot of things. You know, you need a high density camera, processing power, whatever, lot of factors. Actually, the one limiting factor is that when you go, every pixel is an application of current. So when you have 30 by 30, the electrode has certain size, you know, because retina is only so much, you know, 4 by 4 millimeter or something. So the current gets very concentrated. So you make the smaller the electrode, the current gets even more concentrated. So in the end, it will burn the tissue. So it's a limiting factor. So getting around that is a serious problem. So there are a lot of research being done on improving the electrode technology from traditional metal electrodes to, you know, carbon nanotubes or even certain optical methods. So that. Now coming back to your point, yes, anything and anywhere when you stimulate, it's like if you run, I'm stimulating, I get tired, right? If I have a muscle stimulator, it's going to go into fatigue. It cannot be. Cardiac muscle, it turns out, doesn't fatigue. So there it's a bit of an exception. So the neural, it's the same way, but fatigue means you're overexciting or depleting neurotransmitters or something. In rods and cones, you may be doing something else. Maybe excess current injection can be harmful. So you might just turn on the retinal imager sometimes to save power also, because the implant has only so much power. Afterwards, it'll gone and you'll need another one. Current cost is about 100,000 minimum and then additional cost. Okay. Another quiz question. Okay, quick. How many for A? B? C? D? E? Okay, so good. All right, so it's E. Yeah? Okay, so these are some sort of research questions. These are things, take a picture if you're curious. Now listen, you can't research all of these tonight, although you may, that'll be great. But you know, I'm saying that it's something that based on your curiosity, topic that interests you. Maybe you want to research emotions and we can have a discussion at 8 p.m. on that, or 6 p.m. We can, some of you are very curious about deep brain stimulation and so on. You know, one of you want to invent one or develop one for India because it's more affordable or something. So number one, the first one definitely is kind of interesting and highly relevant to what we discussed. Now the second and third, I'm kind of pushing you even a little bit further. It's nothing to do with anything I've introduced so far, but like, you know, sometimes you see a breakthrough that's in front of you, you know, and so this is likely a potentially future Nobel Prize. So we think of stimulation, as we spoke so far, as electrical. It always seems like electrical. But what I should mention is that actually you can stimulate brain magnetically. How many are electrical engineers here? Any electrical? Okay, so I'm going to pick on 3 or 4 of you. Okay, so how does magnetic stimulation lead to electrical stimulation? What is the physics? What's the law behind it? You are a physicist, right? Oh my God. If you get it right. Okay, go ahead.
Sir, we put magnetic stimulator, then it may work on surface or deep. When magnetic current is producing in coil, then in knots it creates electric current in parallel, like this. If magnetic current is this way, then electric current will produce in this way.
Pretty good. So what is what is that law called?
Sir, I don't remember it, but right hand.
No, that's Faraday.
Faraday or right.
No, but he's talking about that law.
Sir, there are 2 laws. I don't remember its name.
That's Lenz's law.
So it's Biot-Savart law, but then that's the orthogonal, because that's what he said. Although magnetic induction is occurring, so it's great. Yeah. So see, some of you took physics, forgot it. Some of you took electrical engineering, didn't connect. But yes, so magnetic stimulation is that a high level of magnetic field is produced by passing current into a coil, right? So that means that right-hand rule. So it's, you know, so that means that current will produce magnetic field, which in turn, going back to this induction, it induces the current. So I put a coil on the head, which produces a huge, you know, it's a very high current, like 1 or 2,000 amperes. When that current is produces, it produces the magnetic field, which in turn produces reciprocal induced current, and that stimulates. So it's not easy to focus, but hey, it's non-invasive, so that has some advantage, and it's used for certain you know, clinical problems. So that's valid. Very recent areas, people are starting to look at ultrasound as a way to stimulate, which is kind of rather unexpected, because people have used ultrasound for actually imaging or for treating things like kidney stones and all that. And instead now with low level ultrasound you can actually directly stimulate. So it's kind of like very emerging. And then this one, optogenetics. So it turns out, you know, like things, certain jellyfish, those things, they are light receptive, right? They produce light. There's certain fish, certain low level creatures are photoreceptive deep inside the ocean and so on. So they found that it's the same way that we have in our eye, light sensitive receptors, right? They are light, they convert light to electricity. So how does that light to electricity, you know, conversion occur? So these are opsins. These are proteins that convert light energy, which bumps up an electron and all of that and creates current. So by going around and looking these kind of, one European person who might be the third person to join this is discovered that certain these proteins exist in these kind of like, I don't remember exactly, but some jellyfish or some equivalent of some very small lower level invertebrates, and they purified that protein, and it's kind of like light produces current. So then these two guys, that's what the professor and Boyden was his postdoc, and they, I think PhD student, I don't remember, they said, oh, let's try that in neurons. So there's a certain protein called channel rhodopsin, anyway, they put it in the neuron and then shone the light, and ta-da, the neuron can get excited. There's another protein called helorhodopsin, you stimulate the light, the neuron stops firing, neuron is not active. So now with light, you can turn a brain cell on or off. Wow, optical communication to this. But it's so pinpoint because you can target individual cell, laser beam and so on. So it's a fabulous now a research tool. So you can look up some of these things, but some people may should think, oh, maybe I can make a retinal processes that way, because by light directly to that rather than electrical current and so on. There is one problem right now, that they need a virus to deliver that protein. So that's a problem. Medically, clinically, you don't want to get virus because you don't know how they're going to work. But I mean, it's an emerging area. There has been actually a clinical study approved, so it might happen someday. I talked about spinal cord injury, right? So stimulation of spinal cord for locomotion. So if you look up this guy, Cortin, and you'll find some wonderful videos. He has done stimulation of the spinal cord in a rat and monkeys to make it walk, and so that's a great example of spinal cord stimulation. So of course, that's spinal cord. Maybe there are a bunch of other therapies and treatments you can research. So follow your curiosity, and who knows. So that's my lecture 2. All right, so when a lecture ends, what do? Everybody get, oh, no. You get up, you stretch, do some deep breathing, talk to your neighbor about some of these provocative topics. I get a little, catch my breath, let's relax a little bit. Okay, so a few minutes, do whatever, go to the bathroom, whatever you want to do. Okay, so let's meet in about, I don't know, less than 10 minutes. But those 10 minutes can be used in a relaxation or interaction. Okay, all right, so come back in 10 minutes.